speaking about the synapse last time, and we left off by speaking about the events that take place on the axon terminal of our presynaptic neuron in order to transfer that chemical signal to that second neuron at the synapse. So that's where we're going to pick up today. Okay, so the synapse. Okay, and we spoke about two neurons, that first neuron being the presynaptic neuron, and then the second neuron in question being the postsynaptic neuron. Now we are going to speak about different options for this second cell, but for right now we're going to consider it to be the second neuron, and that would make this region the synapse. Okay? And so we can describe the space that's between the neuron and its targeting cell or neuron as the synaptic cleft. Okay, we'll speak about some important features of the synaptic cleft. So let's speak about what happens here. First, let's talk about some of the structures. So you know that there are synaptic vesicles inside the axon terminal of this first neuron. So this is our axon terminal. And we've got some synaptic vesicles. And inside those synaptic vesicles is our neurotransmitter in question. So whatever that is, there are a few options which we'll speak about, but whatever that neurotransmitter is, it's located inside those synaptic vesicles. Also want to point out the different channels that are associated with this first neuron. So along the axon, like this is the axon, we spoke about myelination and we spoke about the concentration of what type of channels in between those sheets of myelin. What type of channels did we say were in between these little gaps that facilitated the action potential jumping from node to node, those nodes of Van Baer? So these are voltage gated sodium channels, right? And so we had the action potential going from this spot to this spot until it gets to the axon terminal. Now there's a different channel at the axon terminal, right? These are voltage gated calcium channels. Let's draw a couple of them on. Okay. And so when the action potential gets to the axon terminal, we basically depolarize the axon terminal, and that triggers the opening up of these voltage-gated calcium channels, and calcium rushes into this axon terminal. So we've got calcium rushing in here, calcium rushing in. Okay. Now, when calcium rushes in, this facilitates or triggers the exocytosis of these vesicles. So these vesicles will migrate to the membrane. They'll dock, their membrane will fuse with that of the axon terminal itself, and then we'll dump all of this neurotransmitter into the synaptic cleft. Okay. Now, there's a very specific receptor that's located on our response cell, on our postsynaptic neuron. And so that receptor, let's draw a receptor here, is going to specifically bind our neurotransmitter. Okay. Now we'll talk about some different options for this receptor in terms of bringing about a response, but binding of the neurotransmitter to this receptor is what brings about a cellular response. And we'll go on today to speak about fast versus slow responses here. Okay, so we won't get into too much detail right now, but later on we're going to differentiate between a fast response here and a slow response here that is brought about by the binding of this neurotransmitter to that receptor. Right? We can have different types of receptors. Now, that's going to transfer this electrical signal to chemical signals, which cross the synaptic cleft, and then we can reconvert this chemical signal back to an electrical signal on the postsynaptic side. But at the end of this event, at the end of this signal, we need to clear the excess neurotransmitter from the junction. Otherwise, we will be sending this signal indefinitely. So this is a really important part of synaptic communication, clearing away the excess neurotransmitter. And so we have two important structures that are on the that are at the synaptic cleft that are responsible for clearing this excess neurotransmitter. One is called this enzyme. This is called acetylcholinesterase. Okay, it's a really long name. We'll go on to speak more about it, but basically it's going to degrade this neurotransmitter, which is usually acetylcholine. And this enzyme will break this neurotransmitter down to smaller pieces, and then we can have another structure here. So we've got a third structure, this reuptake molecule, that is taking this neurotransmitter, breaking it up, and recycling it back into the previous cell, back into the axon terminal. Okay, so let's go over these sequence of events one more time. So we've got the action potential moving along the nodes of Ranvier. It's jumping down the length of the axon. We describe that as being saltatory conduction. When the action potential gets to the axon terminal, we no longer have voltage-gated sodium channels here. Instead, we have voltage-gated calcium channels. And so when the membrane potential changes at the axon terminal, these voltage-gated calcium channels are opened and calcium rushes into this axon terminal. That then triggers the exocytosis of this vesicle. It's like that word here, actually. Right, so that's right, exocytosis, which is basically the dumping of this neurotransmitter into the synaptic cleft. That will cross the cleft and bind to a specific receptor, which will bring about some type of response on this second cell. And then in order to cease this communication, we have an enzyme which degrades the excess neurotransmitter. And then that neurotransmitter can either be diffused away or it can be reuptaken via this reuptake molecule, recycled and repackaged to make a new synaptic vesicle. Okay. All right. Any questions on that sequence of events? Any questions? Make sure you understand this in detail. Make sure you're able to recreate this, that you know the steps that are happening. We're going to go over the steps here in a bit. But basically the steps 1 to 7 of how this signal is relayed. Okay, so this is just a review of what we just spoke about with some detailed steps indicating each of the events here. And so the action potential we know comes down the axon. It opens up those voltage-gated calcium channels. This triggers the docking of that vesicle to the axon terminal membrane, which allows that neurotransmitter to diffuse across, bind to its receptor, which is here, and that will then bring about some type of cellular response. Okay. Now, in order to terminate this response, we must clear the excess neurotransmitter. So you can kind of see that happening here. We've got this enzyme that we talked about that degrades the excess neurotransmitter. And then we can also diffuse it away in its degraded form. We can diffuse it away in its whole or in its full form, or we can reuptake it back into the previous neuron to be recycled. Okay, so there are a couple options for clearing the junction of that excess neurotransmitter.